Coming to understand digestive system in man and digestion process in man, so we have the digestive system to be divided into two main distinguishable divisions. So that is nothing but the alimentary canal. So alimentary canal will be the passage which allows the food to pass through it. So that will start from the buccal cavity or the mouth here actually, which is the oral aperture through which ingestion of food can be done. So mouth leads into the buccal cavity leads into the esophagus to stomach is continued as small intestine, and small intestine will be continuing as your large intestine, and then we have the rectum, which will be the temporary storage for food, undigested one, and then the digestion, which is done through anus, which is the aperture through which undigested food is thrown out. So this passage is what you call it as the alimentary canal. Then we have the associated glands which will help them in digestion, and those are main three glands we will have, that is the salivary glands which are present at the buccal cavity or in the face region or in the mouth region, and then we have the liver which secretes the bile juice, and we have the pancreas. Along with that, we will also have gastric glands in the walls of the stomach and intestinal glands in the walls of intestine. Now coming to understand the process of digestion. Digestion in case of human beings is of two types. One is mechanical digestion and another one is chemical digestion. So in case of mechanical digestion, what happens is digestion takes place by chewing process, as in case of chewing by teeth, and churning process, as in case of stomach here, highly muscular J-shaped organ it will be. So this will take up churning of the food in order to make it a paste here. Okay, so that is what you call it as mechanical digestion, which takes place by mechanical action of the organs which are present. Then you have chemical digestion, which is a type of digestion wherein the food substances will be broken down from complex compounds into simpler forms by the action of enzymes. So that wherever digestion is taking place by the action of enzymes, you call that type of digestion as chemical digestion. Okay, now let us understand how digestion will take place in case of humans. Once the food is ingested through the mouth here, the chewing action takes place by the help of teeth. So teeth will help in chewing the food and breaking them into smaller particles. So that's the mechanical digestion here. So chemical digestion also starts in the mouth, and here chemical digestion of only carbohydrates is initiated. So that is taken up by the salivary juice here. Okay, so salivary juice is secreted by salivary glands. In humans we have three pairs of salivary glands. So one pair is called as the parotid glands, P-A-R-O-T-I-D, parotid glands. So these parotid glands are the salivary glands which are present near the ears. Then you have the sublingual glands. So those are the pair of salivary glands which are present sublingual. Sub means below, lingual means tongue. Then we have submandibular salivary glands, which are nothing but the salivary glands which are present near your jaw region, below your jaw region. So those are the three pairs of salivary glands. All these three pairs of salivary glands will secrete lot of saliva. So saliva, it's a alkaline juice which comes in contact with the food in the mouth. So there are many functions of saliva, but related to digestion we consider two main functions of saliva. One is saliva will make the food moist, and it will help in binding the food in order to make it into bolus, and also makes it helps it in lubrication of the food here. Got it? So then here you have very important enzyme in saliva which is called as salivary amylase, S-A-L-I-V-A-R-Y, salivary A-M-Y-L-A-S-E, salivary amylase, or otherwise it is also called as tallin, P-T-Y-A-L-I-N, tallin enzyme. So this enzyme acts on the carbohydrates in the food here. So that carbohydrates are nothing but starch. We consume lot of starch, and starch partially it is digested into maltose here. So once the completion in the sense whatever completion of carbohydrate digestion takes place here, that is a partial digestion when it takes place here, the food is swallowed in the form of bolus. So bolus is nothing but B-O-L-U-S, bolus is nothing but the lump of food which is swallowed through the esophagus here. So esophagus is a long tube which connects the buccal cavity with the stomach. So esophagus doesn't have any digestive function but just acts as a passage for the chewed food from the buccal cavity to the stomach here. Why there is no digestion? Because there are no digestive enzymes secreted in this long tube which is esophagus. So once the food enters into the stomach here, if the food has to enter here, at the anterior end as well as the posterior end of the stomach, you have two sphincter muscles. One is called as the anterior sphincter or esophageal sphincter you call it as, and another one at the end of the stomach you will have which is called as the posterior sphincter or the intestinal sphincter muscle, which is present at the junction of stomach as well as the intestine. So here food, when it enters into the stomach, the sphincter muscles lock up the food in the stomach in order to help digestion. So here first mechanical digestion is done in the stomach by the peristaltic movement. What is peristaltic movement? Peristaltic movement is a rhythmic contraction and relaxation of the digestive system muscles or the alimentary gut muscles, which helps in movement of food. Here peristalsis in stomach helps in grinding the food from a complex form into a smooth form like a paste like thing. Got it? So then here there is action of chemical enzymes also. So the chemical enzymes are nothing but the secretions of the gastric glands. So overall secretions of gastric glands are called as gastric juice. So that gastric juice will contain an acid. The acid which is secreted here is hydrochloric acid. So hydrochloric acid will have two main functions. What is the main functions of hydrochloric acid? It is a 2 marks important question again. So first function of hydrochloric acid is, if suppose any microbes have entered into the stomach along with the food, the acid will kill those microbes and eliminate them. Then the acidic content which is present here, hydrochloric acid, will help in activating an enzyme which is secreted by stomach or the gastric glands of the stomach, that is nothing but pepsin. So pepsin is a protein digesting enzyme, but pepsin is not secreted in the form of pepsin itself. It is secreted in an inactive form called as pepsinogen, P-E-P-S-I-N-O-G-E-N, pepsinogen. So in the presence of acid, pepsinogen gets converted into pepsin, which is an active protein digesting enzyme, and the enzyme will act on proteins and break them down into polypeptides. Again here you can understand it is only partial digestion of proteins. Proteins are digested into polypeptides. No other food components are acted here, only proteins are digested. Along with gastric glands, the walls of stomach will also have very important glands which are nothing but the mucous glands. So this mucous glands, as the name itself tells you, will secrete mucus, which is a very slimy substance. So this mucus will help to make the food lubricated so that easily it can pass, and as well as it will protect the walls of the stomach against the action of acid, hydrochloric acid, which is a strong acid here. So once the completion of digestion of food takes place in the stomach, it takes about 4 hours for the completion of digestion in stomach. So and posterior sphincter will open up and food is slowly passed at intervals to the small intestine. When first food enters into the small intestine, it comes in contact with the secretions of the largest gland of our body, which is nothing but the liver. Liver secretes bile juice. So bile juice is, once it is secreted, it will be stored in the gallbladder, which is a tiny organ here, which is green in color usually. So usually it will store here and then it is secreted. Sometimes it can even directly secrete it to the small intestine also. So bile juice is not having, it doesn't have any specific digestive function, but it takes up a very important function in digestion. So what are those functions? Again, what are the functions of bile juice will be an important 2 marks question here. So first function of bile juice is it takes up emulsification of fats. You can remember till now fats have not been digested. So first process which acts on fats is it will emulsify, not digestion, it will just emulsify. What is emulsification? Fats which are complex substances will be broken down into simple or smaller particles, they will be broken down and their surfaces will be made smoother for enabling further action of fat digesting enzymes on them. So that is what you call it as emulsification. The surfaces will be made smooth in order to enable the enzymatic action. Second very important function of bile juice is that, see, once here the chyme, after digestion, the paste of food what is passed on further, it is called as the chyme, chyme, chyme. So this chyme, until it is there in stomach, it will be acidic in medium, but this chyme, when it comes into intestine, compulsory it has to be turned into alkaline medium or basic medium. Alkaline medium it has to be turned, because unless it is turned into alkaline medium, the secretions of pancreatic gland or pancreatic juices cannot act on it, and intestinal juices also cannot act on it. So hence it's very important that the medium of the chyme should be converted from acidic to alkaline or basic. Got it? So that is done by the bile juice here. So you know, bile juice contains lot of sodium bicarbonate, NaHCO3. That sodium bicarbonate will enable turning off the acidic medium of the chyme into alkaline medium for further action of enzymes. So once that is done, then pancreatic secretions will act on the food here. So pancreatic secretions contain, or pancreatic juice contains all three types of enzymes. So pancreatic amylase you have, again whenever I tell amylase, it should be understood that it's a carbohydrate digesting enzyme. Pancreatic lipase you have, and pancreatic proteases also. So lipase is an enzyme which digests fats, and proteases are enzymes which digest the proteins. So these proteins, pancreatic proteases, are particularly two very important proteins you have to, enzymes you have to remember. So those are nothing but trypsin as well as chymotrypsin. So all these will act on it. Now some of the carbohydrates which are digested into maltose here will be acted upon, and some of the starch will be left over here without digestion also. So that will be acted upon by pancreatic amylase, and it will be converted into maltose and glucose, and pancreatic lipase will act on the emulsified fats and break them down into fatty acids and glycerol. Then the pancreatic trypsin as well as chymotrypsin, here whatever proteins were digested into polypeptides, they will act on that and then take up protein digestion. Undigested proteins will be broken down by trypsin and chymotrypsin into polypeptides, and polypeptides into amino acids. So once it enters into the intestine here, further intestinal glands which are present in the walls of the intestine will be secreting intestinal juice. So similarly, intestinal juice also contains all the three types of enzymes: intestinal amylase, then you have intestinal proteases and intestinal lipases. So same here, maltose will be converted into glucose by the intestinal amylase. The polypeptides will be broken down into peptides as well as amino acids, and which are the absorbable forms here, and the fats, undigested fats, will be broken down into fatty acids and glycerol. So you know, here there is an very important question is that how is our small intestine modified in order to take up absorption of food. So here in case of intestine, the walls of intestine will have finger-like projections which you call them as villi, V-I-L-L-I, villi. So these finger-like projections will increase the surface area of the small intestine by about 8 times. So this increased surface area will provide a lot of surface area for the absorption of food. The inner space of villi will also contain a space which you call it as lacteal. So that lacteal will contain a connection of your artery, vein as well as the capillaries, which will help in absorption of your fats. Got it? So once the absorption of food is done here what happens is the food, carbohydrate digested food which is in the form of glucose, will be taken up by the blood vessels which surround here. I told you by the third step, which is called as absorption of food, glucose can be absorbed and amino acids also can be absorbed by the blood, but fatty acids cannot be absorbed by the blood. It is absorbed by the lymph, which is the lymph connection which will be found in the lacteal. So lymph will take up fats or fatty acids along with fat soluble vitamins, which are nothing but vitamin A and vitamin D. Those will be absorbed by the lymph here, which is a colorless blood, and it flows through the lymph vessel, whereas blood will absorb and transport glucose, then amino acids as well as other vitamins which are water soluble vitamins, which are vitamin B and vitamin C. So once absorption of food is completed here, the food will be undigested food which is not necessary for the body will be moved into the large intestine here. Particularly, there is no process of digestion in large intestine as digestion is completed in the small intestine. So here only excess amount of water is reabsorbed into the body. Here further food will undergo fermentation by the action of bacteria which are present in the large intestine. So here what happens is the bacteria which acts on the food will produce vitamin B complex, B1, B2, B3 and B12. So those will be used up, vitamins will be used up by the body here, and food, when it is undigested food which is solidified by the absorption of excess amount of water, will pass through this large intestine and it comes to a temporary storage part which is called as the rectum, RECTUM, rectum here. So, and when it is time for throwing of the food, it is thrown out through the anus, which is the opening through which undigested food is digested out of humans. So hope you have understood how digestion will take place. Now we will have a recap of the enzymatic actions, where what will happen and how the food gets digested. So this is a recap of your action of digestive enzymes, children. So first, starch. It comes in contact with salivary amylase or tallin. We learned that PTYALIN, tallin. It is just converted into maltose, which is a disaccharide. And further, this maltose comes in contact with amylases, which are nothing but your pancreatic amylase as well as the intestinal amylase, which break them down into glucose. You have other carbohydrates also which are acted upon by particular enzymes. For example, sucrose is acted upon by sucrase and it is converted into glucose, and lactose, which is a milk sugar, will be acted upon by lactase, which is again an amylase, and it will be broken down into glucose. So then coming to proteins, they are acted upon first by pepsin in the stomach. So pepsin partially digests it into polypeptides, and again polypeptides come in contact with general proteases. So proteases are the protein digesting enzymes. So you have the protein digesting enzymes in case of pancreatic, trypsin and chymotrypsin, as well as intestinal proteases, which finally break down these polypeptides into amino acids, which are the final products of your protein digestion. And coming to fats, fats are first emulsified by bile juice, then they are acted upon by pancreatic lipase as well as intestinal lipase, and the final product of fat digestion will be fatty acids and glycerol. So these are the forms in which food can be absorbed. Carbohydrates are absorbed in the form of glucose, and amino acids are absorbed, proteins are absorbed in the form of amino acids, and these are taken up by blood from the intestine, and fatty acids and glycerol as well as fat soluble vitamins are taken up by the lymph, which is the colorless blood present in lacteal, which is the space which is formed in villi. So that's it, children. Hope you have understood the process of digestion in humans. Thank you for listening.
ನಾವು ಮಾಡ್ತಾ ಇರೋ ವಿಡಿಯೋಗಳು ನಿಮ್ಗೆ ಇಷ್ಟ ಆಗಿದ್ದಲ್ಲಿ ಲೈಕ್ ಮಾಡಿ, ಶೇರ್ ಮಾಡಿ, ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡಿ. ಪಕ್ಕದಲ್ಲಿರೋ ಬೆಲ್ ಐಕನ್ ಅನ್ನು ಕ್ಲಿಕ್ ಮಾಡಿದ್ರೆ ನಾವು ಮಾಡೋ ವಿಡಿಯೋಗಳ ನೋಟಿಫಿಕೇಶನ್ ನಿಮಗೆ ಮೊದಲು ಬರ್ತದೆ. ಯಾವುದೇ ರೀತಿ ಸಂದೇಹಗಳಿದ್ರೆ ಕಮೆಂಟ್ ಸೆಕ್ಷನ್ ಅಲ್ಲಿ ತಿಳಿಸಿ. ಹಾಗೆ ಈ ತರಗತಿಗಳು ಹೆಚ್ಚಿನ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ತಲುಪುವಂತೆ ಶೇರ್ ಮಾಡಿ.